All right, I have 12 noon here in College Station, Texas, and folks back east or should be around 1 o'clock. This is Greg Pompelli. I am the voice that you can't see, but that's better for everybody's video. I'm the director of the Cross-Border Threat Screening and Supply Chain Defense Center of Excellence, DHS Center of Excellence here at Texas A&M. And today we have a wonderful opportunity to hear from Dr. Michael Pravica, who is a professor in the physics department at the University of Nevada, Las Vegas. He does most of his work, or had done most of his work, in high pressure science with his PhD from Harvard and undergraduate from Caltech. So we were very lucky last summer when he applied to get one of the summer research team program projects that was successful, and so successful that it then led to follow-on work by DHS or with DHS in this area of kind of non-destructive detection of pathogens in particular. So I think it's wonderful to have him here. I think one of the things that you're going to find as you hear him today is he's passionate about what he does. So when I say that Dr. Pravica is an extremely inspirational and passionate physics professor, that it's important to attend his lectures. So pay attention as you're here today. He also grades fairly, and he's accessible outside the lecture. So I'm going to translate all of that because those aren't my words. Those are words that have come from his students. And but hopefully you'll find that if there's something of interest, he's going to be open to talking to you. And you don't need to go through our center to get to him because he's a wonderful resource for everyone. So, and if finally, just to let you know that he's not camera shy, if you haven't seen his work on all birds with Marshawn Lynch to talk to students about science and climate change, go to YouTube, put in, I guess, all birds and Marshawn Lynch, and you're going to see some really funny but also insightful educational videos that were developed where Michael is absolutely key. So with that, I'm going to hand the mic over to Michael. But before I do, I'm just going to let you know that if you have questions, you're certainly, Mike's willing to take them on as we're going. But please, if you turn your mic on, remember to turn it off. And then also as you're, if you want, you can hold the questions toward the end. So we're recording this, and the hope is that we'll be able to get this up on our website within pretty short order. So thank you for everyone for joining us, and Michael, the floor is yours.
Thank you, Greg, for inviting me. And again, I do apologize that I'm not able to be there in person due to having suffering a knee fracture. But I am excited to give an update on sort of what we've been doing related to cross-border threat security. And I'm going to talk about developing a spectroscopic detection strategy and possibly neutralization strategies for biological threats crossing our border. And so there are really kind of just 2.5 components I want to talk about in this talk, continuing our development of a hybrid Raman UV vis spectrometer for rapid detection of certain threats. I have at least one idea that I garnered when we did a visit to the San Ysidro and Otay Mesa border facilities. So I want to talk about that, and then briefly, and then talk about some ideas that have been evolving pertaining to eradicating immediate threats that might be discovered by officers at the border. And so just to give you a little bit of a background of what we did last summer and the summer before, particularly the summer before, we were looking at 4 types of threats: viral, bacterial, mold, and then chemical, which would include, things like ibuprofen, acetaminophen. We used threats that were not really threats because I'm not a biologist and I don't have hazardous biohazard training or facilities in my lab. But we used, things like the tobacco mosaic virus, which you can order online, E. coli, K12 bacteria to kind of be representative for bacteria, and then yeast for something that might be like representative for a mold, and then just simple drugs like acetaminophen, ibuprofen, and nicotine as chemical poisons. And so the idea was that we were looking at different strategies, and in our I'm a spectroscopist, so I am very wedded to the ideas of non-invasive electromagnetic based types of detection methods, such as NMR, Raman spectroscopy, and other optical or even infrared techniques. And but of course, there are other methods that are used, which I call active methods, which would be like the PCR tests, where you're actually reacting in the presence of a solvent, your substance in question, and then doing some sort of chemical analysis after post facto to kind of basically see what it is. I was interested in something that was minimal intervention, no chemical alteration. So we were looking again at some sort of a spectroscopy like Raman or infrared, which I call passive spectroscopies. And there are, of course, related to the active techniques, there are some beautiful devices that have been set up. For example, Los Alamos set up the Pegasus biosensor in the lower left here. There are field deployable systems here for biosensing, a whole bunch of articles. And so we kind of sort of did a study of these different detection methods, which are great, but of course, these machines tend to suffer from anything that has moving parts, like a centrifuge or pumps, will tend to wear out. And then I can tell you from personal experience as an experimentalist, if something can go wrong, it inevitably will. We call that Murphy's Law. And so I was looking at something more solid state, something that wasn't going to have all sorts of solvents flowing and detection processes. So what we were looking at was the following, what I call boundary conditions. Does the interrogation method damage the sample? Is it dangerous to the operator? How much training would be required for the operator? How long would it take to perform a test and get a positive, get some sort of a result? Of course, expense is always an issue, somewhat. Reliability, ruggedness of the equipment. Can it handle high temperature inside a trailer or whatever other conditions, rain, for example? What about the possibility of remote detection? Anything where you reduce the chance of an officer touching a poison like fentanyl, for example, would be a very positive thing, because if they could do that remotely, because that would give them a layer of separation. And then, of course, what kinds of reagents, chemicals, solvents, how many repeated measurements do you need to get a consistent answer? And basically what I would call supplies. What happens if in the supply chain you're not able to get some reagent, and do you have stocks for this, and how do you plan those kinds of things? And then, as I kind of implied earlier, with the reliability, measurements in extreme conditions are always of interest. So in those kinds of spirits, and as a physicist, as a spectroscopist, I sort of decided to look into passive spectroscopic methods for rapid testing. And let me just say again at the outstart here, what was about this interesting, but what's been interesting about this odyssey is, you think you can solve all the world's problems, but you can't. So you start in this big vision, and then you say, let's kind of just hone it down a little bit into something that within the limitations of time and finance, that we can actually get something that might be useful for at least something that would be of interest to the DHS. So of course, as I said, as a physicist, much of everything can be boiled down to waves and particles. And in particular from the waves, most of what we have to deal with is the electromagnetic spectrum. So I'm mostly interested in the optical range, and then I'll talk a little bit about the X-ray range. Um, there are techniques. Infrared is very powerful. The only problem with infrared is that if you're dealing with a hot trailer, you get a lot of overwhelming noise in your signal. So that's something you have to deal with. And so, again, just this is just a brief intro to what we did 2 summers ago. We did Raman spectroscopy, UV visible, NMR, cyclic photometry, and fluorescence spectroscopy. And Raman, of course, that's really mostly my specialty. It's powerful. You know, you can see right away. Oh, I've got aspirin on top. Uh, I've got Tylenol, acetaminophen second from the top. I have ibuprofen third from the top. These are very standard signatures. We call them chemical signatures that basically are associated with vibrations of bonds within these molecules. And then the lower trace, two traces, you can see Raman spectra of various mixtures. And it was exciting for us to find that there is a lot of literature, there's a lot of devices out there that are portable that can deal with 2 or 3 mixtures of powders. And so that was very powerful for us to see that firsthand, how powerful this can be to decipher what's inside some sort of mixture of unknown powders. And then we looked into UV vis absorption spectra, and as you can see, looking at acetaminophen on the upper left, E. coli in the upper middle, uracil, constituent of RNA in the upper right, and then the tobacco mosaic virus, lower left, tortilla yeast RNA, lower middle, and then herring sperm DNA. These signatures all are quite, have differentials. And one of the interesting things just to kind of note is that you can see the tortilla RNA, and the E. coli in the middle, they have a longer tail. And so what happens is that in living systems, they're very coupled with each other. So they share energy, all these molecular systems. And so what that implies is you have usually a very broad spectrum. And so that was something we realized, oh, actually, this would be a very powerful technique, UV vis absorption, just to basically identify, you see something, is it chemical? Is it living? Like, or is it viral, like anthrax? And so right away you could see, yeah, I can differentiate from the acetaminophen in the upper left to the E. coli bacteria because I see this very long tail. And so, and that's something that I think would be very powerful for officers. And again, the idea is a quick split minute, whatever decision. They see something strange. Is this an immediate threat? Is it living? Is it chemical? At least they could, with UV vis absorption spectra, they might be able to determine, oh, that's something living, or that's just chalk. Okay. So, and NMR, of course, is, which came from physics also, is probably the most powerful technique in terms of being able to differentiate because it is sensitive to the local magnetic field applied to a proton or other NMR active nucleus. So it's very pristine. You get very sharp peaks, and you can say without, beyond the shadow of a doubt, this is, such and such chemical and whatnot. The only problem with NMR is it usually requires very low temperatures and, people and a high level of skill to operate the spectrometer. I'll talk more about that a little bit later. So just to repeat what we concluded from 2 summers ago was that our approach was that Raman is the quickest way to determine if something is, let's say, chemical poison. Then the next step might say, okay, if it's not a chemical poison, what about, is it living? Is it something like a bacterium or virus? Then UV vis, looking at that long tail, might give you a quick, okay, this is something that might have a good chance it's living, and therefore we at least understand some aspect of the threat. Then the next step would be to go to NMR. NMR spectra can be taken very quickly. Usually, unfortunately, you have to put them in a solvent. You spin up something or put it in a magnet, and then you get your NMR spectrum. So that might give you more information that would say, well, is this really something we need to worry about? And then you go to the active techniques to basically looking at PCR tests or whatnot to get a further handle on what you're seeing. So then again, we had proposed a setup where we had some sort of a combination of a Raman and UV vis. The issue of Raman is it's very narrow in terms of the frequency spectrum basis. So you might excite something with a 514 nanometer laser, and you're looking within a few, maybe 10 nanometers within that 514 for what we call Raman inelastic scattering. In the case of UV vis, it's much more widespread. So you're looking maybe from something like 350 nanometers all the way down to maybe 800 nanometers. So it's a wider part of the that spectrum. And the only issue is that it's a little longer usually to acquire those, and it's not as it's harder to to get a definitive like, for example, if you have a chemical, exactly what it is, because you're not looking at specific bond vibrations. You're looking at a coupling of energy within the molecule within that whole range of the electromagnetic spectrum. So the key is, if you're going to do both, you'll need a grating within the spectrometer, a movable grating, something that you can disperse the light very narrowly in the case of Raman, or disperse it much more, or I'm sorry, other way around, disperse it much more widely in the case of Raman, and then disperse much less so you can get much more of the spectrum in the case of UV vis. So, and we found these portable spectrometers and that have their own libraries. And again, and I'll talk more about this when we made our visit to the DHS facility at San Ysidro, and saw something similar to this. So they're out there, but again, the point is to, can we do a combination of techniques right there in the field, in that, in the trailer truck, to right away determine, without having to go back to a lab, that is this living, is it some sort of fentanyl chemical toxin, at least to get a little bit of an answer for the safety of the officers. And then, of course, there are also portable UV vis spectrometers, which are cheap, relatively cheap. And also, they have their own chemical libraries. So it's kind of was exciting, like, oh, we can actually do both of these techniques. And again, given that we've seen the power of both techniques to sort of at least differentiate between certain different types of species of or types of threat. And then the portable benchtop NMR spectrometer would not be something that would be right there in the field, but it might be in the lab at the facility. And the coolest thing that has happened in the past 20, 30 years is that there are portable NMR spectrometers that you do not need liquid helium, which is going through the roof in terms of the price and scarcity, and literally going through the roof. So the thing is, this would be a powerful method, again, as a initially doing initial tests to get some further information on the threat. So what we did as part of our grant was as again, I'm an experimentalist, so everything I have to do, I want to be in the field. I want to be in the lab. I want to see how people do things. And so we visited two of the facilities on the California-Mexico border. We went to this San Ysidro, which is basically connects with Tijuana in Mexico. And we went to the Otay Mesa DHS border facility. So the San Ysidro is mostly used for passenger traffic. They do have some limitations. You could take a small, I don't know, a van or a truck or something. But basically, the Otay Mesa is where the commercial trucks go through. And I actually walked the night before my visit. I walked into Tijuana. I had my passport card, and I kind of want to test it out, and had a very nice visit there because I had a hotel right by the Otay Mesa, and so I got to see some of the facilities, took a picture here and there at nighttime there, and we were very blessed with Officer Daniel Yale, who is the agricultural operations manager, and I think he had just been promoted to something else. I kind of forget now, but wonderful treasure trove of information, and as far as what kinds of challenges the officers face on it. And we went there, so we watched the officers in real time. My student Drake on the upper right went with me. Getting there was quite fascinating. Well, we actually met him at the San Ysidro crossing. So they have all these interesting, kind of like a maze, ins and outs that you can kind of get in there. So we had to take a U-turn on the lower right to get into the facility. He's like, don't miss that U-turn because then you're going into Mexico. So everything worked out well. And again, part of my desire for this talk is to somehow articulate how important it is that if you want, problems solved, that the scientists, engineers, technicians need to be there to see what they're facing. So I just want to give you an example. On the upper left, this was at San Isidro, and these are all magnets that are on some sort of a metal sheeting. I didn't want to they didn't tell me I couldn't take pictures, but I was kind of trying to be like, not to, I didn't know what was, if there was anything classified or whatever. But apparently there's a major problem there where these drug lords will make these packages of drugs, and they'll put in one of those magnets and affix them to the bottom of the undercarriage of some unsuspecting or suspecting person to carry them across the border. And so I was discussing this, with the officers, and I said, well, actually, and these magnets, apparently many of them come from computer, like the hard drive magnets. So I said, well, wait a minute. That means they're very strong, permanent magnets. Why don't you, and I'm watching these cars go through as they're being in an inspection. I said, why don't you just put up a pickup coil in your speed bumps? Because as you get the speed bump, you're getting closer to the undercarriage of the vehicle, and it would be so trivial to measure. You'd see just a spike in voltage as the car passes over the speed bump. And I remember talking to the head of the San Ysidro facility, and I apologize, I forgot her name, but again, another treasure trove of information. And she's like, gee, that's a really interesting idea. Maybe that's something we should pursue. So, it was just being there and seeing all these day-to-day operations. And then, maybe it's just the way my mind is constructed as a scientist, I always see gaps, I see deficiencies. And so I'm like, oh, why don't we try this? Why don't we try that? So there were many, many things that we watched there in real time that would, let me just say, and again, I'm not critiquing anything, but just could use improvement. And so again, that was one of them, using a pickup coil to detect these magnets on the undercarriages of cars so that it would be very easy right away to see what's going on. And so this was part of the, where we were going I remember we went into the lab, and there was a very capable, knowledgeable officer there who was showing us how they use their Raman. They have a portable Raman, and how they measure these. So they were more focused on, the fentanyl threats, which is a big threat. And again, there have been officers who have, how should I say? I don't want to say succumbed, but they were intoxicated by just touching fentanyl. And so it worries me when just watching the operations, that, and again, they're there right there with the threats. And so how powerful it would have been just to be able to, you see something outside. I personally wouldn't want to carry a box of something that was suspect that I didn't know what it was, at least not without checking if it was something like fentanyl in the first place before I brought it into the lab for analysis. So again, my philosophy is anything that is a remote way that from a distance, it doesn't have to be a far distance, but some distance, that I can just optically detect something that could be a threat that some officer might accidentally touch, and then get some sort of an overdose. That I think would be very positive. And again, I didn't see that there, and so these are just some of the suggestions that we noticed why I feel this work would be kind of useful. So again, these were just some sort of pictures from the students from 2 summers ago. I should mention that one of the students, the one on the upper left picture in the middle, Nicholas Pudar, he actually graduated and he's now in medical school at Rutgers. So I was excited for Nicholas, but I was bummed out because I was kind of hoping to have him over this year. So we have another student helping us. But this is where some of our measurements in the upper right, we were doing NMR with Jung Jae Ko, NMR technician. On the lower left, we were doing cyclic voltammetry. And then the upper left, we were doing some solvent, working with solvent extraction, drawing things so that, to basically do some chemical analysis. So right now, as it stands, I think the best option is to use a portable Raman UV vis system similar to the ones that I have mentioned. The idea would be that the officer would have this strapped, and they would have either one or two fiber optic cables. And we've developed kind of an optical to compression technique to better collect signals. And so basically from a distance. So let's say there's a crate in some trailer at Otay Mesa. The officer comes up and sees some white powder on that crate, and it doesn't look like anything around the other crates, and just wants to quickly check, is this something living? Is this something chemical? They can use this, maybe it'd be on their strapped on the, on their shoulder, and they can just do a quick Raman just to see, number one, is it fentanyl or something chemical? And then maybe a UV vis after that, or at the same time. And it would be readily, they'd get an answer right, very, they get a spectrum. They might see that long-tailed UV vis, hypercoupled living system kind of a spectrum, and they may, okay, this needs some further investigation. All right, so, and I should mention that there are actually, if you don't mind, I'm going to just, let's see, I just want to, well, anyway, I'll go back, sorry. There are actually techniques, so this is sort of the second part of my talk. Are there any questions before I kind of transition? Is everybody there? Right?
Good. Yes,
Okay.
we hear you.
Okay, good. All right. Okay, so let's, there's no other questions. I'll just go to the next part. I remember being in the San Ysidro facility, and we were talking to the head agent there, and we went in and she's showing us, like, because we were asking all sorts of questions. Well, what happens if you have something contaminated, something with diseased flesh, meat or something? And basically what they do is they sequester, they'll put it into these bins. And it had occurred to me that, gee, if something was really, really dangerous, that you don't need, and then they said they had these contractors that would go in and in some cases burn the material or somehow denature it, bleach it, but they were more private contractors. But again, what does that mean? That's like chemical hazardous waste. I mean, you have to have somebody come in, pick up the material, and then, take it to wherever landfill or wherever the burning facility where they're doing it. And so it just had occurred to me that, again, in the case of something that would be a very serious threat, anthrax or some sort of, botulism, or I don't know, some sort of a really serious hyper contaminated sample, that it would be useful to have like maybe some sort of an irradiation facility. Now, why would I say a radiation facility? Because, high energy electromagnetic radiation is very penetrating and it's ionizing. So basically it's something that would damage this material. The Mail Post Office actually, they use irradiation techniques because they had, I think it was around 2001 or something, there were some anthrax letters that were sent to various people, including, I believe, members of Congress. And so they use, now I had heard, somebody had told me, I'd read somewhere that they use neutrons. Neutrons aren't really a good idea if that's the truth, because neutrons are, first of all, hard to control in terms of energy. And secondly, they tend to, depending on the energy, they can transmute nuclei to make them radioactive. So the best is, as it says in this EPA website, I'm sorry, I couldn't copy the whole website, but it basically says irradiated mail is passed through a high energy beam of electrons or x-rays. Okay, so it sterilizes the mail. So the idea would be to take, if you have some suspect material that is deemed to be a serious threat, is there a way to irradiate with x-rays And X-rays, why? Because X-rays are very penetrating. Those are the X-rays, for example, that found that I had a fracture in my kneecap. So they're very penetrating. But the conventional ways that people use X-rays are typically uh, white, polychromatic, so wasteful energy. So I just want to make a proposition of irradiating immediate biological threats using controlled, selected decomposition with monochromatic hard X-rays. And X-rays are actually pretty easy to produce. You can get them in doctor's offices, dentist offices. They're penetrating, depending on the energy, of course. Higher energy X-rays, like 40 keV, can go well into the human body and therefore food and other contaminants. They can be used in a very tuned manner to destroy particular biological components, thus neutralizing the threats with minimal handling and minimal intervention. So we have a technique that we've been developing, and we actually have this patented, where you can irradiate with a fixed energy, for example, a salt, strontium oxalate, at about 16 keV, just slightly above 16, which is the K edge of strontium. And you can induce chemistry by producing photoelectrons along other, we call it charge transfer. You further ionize the strontium cation, and that induces charge transfer from the oxalate anion to kind of partially neutralize the strontium cation. And for us, we've actually made new materials with this, something called doped polymeric carbon monoxide. And the essence is that basically you're taking advantage of L and K edge absorption of X-rays in a salt, in some sort of a metal cation, to be an abundant source of these photoelectrons, which will then damage whatever is surrounding that cation. And we have some evidence for L-edge induced photochemistry, uh, very strong. And so actually I have an L-edge is just, instead of the 1s, it's the next one, the 2s, higher energy. I mean, so at 1s to like higher level, you're going to the 2s to a higher level. So it's lesser energy, and it is closer to the valence. So it seems to be more destructive. So we've also developed techniques where we've noticed that there are resonances when you use these hard X-rays, basically anything greater than, say, 7 keVs. And just to give you a perspective, 25 keVs of X-rays in terms of wavelength is about a half an angstrom. And that's perfect because that's within the dimensions of atoms and molecules. And so therefore, if you look at bonds as kind of like molecular antennae, then you have an enhancement, just like you would have with the dipole antenna, with these bonds to basically absorb X-ray energy and ionize the bonds and therefore break down the compound. So, for example, pardon me, we can break down easily KClO3, potassium perchlorate, potassium chlorate, into KCl, which is this common light salt, and oxygen. And so you can see on the bottom middle, there's a strong correlation to the energy and the efficiency of the chemical decomposition. So here's my point. If you look at, things like DNA components, the nucleotides, they're all mostly ringed compounds. And I actually have a lot of experience damaging ringed compounds. On the upper left, you'll see triamino trinitrobenzene, which is actually the only explosive, insensitive secondary explosive that's used for nuclear weapons, that's authorized. And to a physicist, it's a beautiful molecule. It's a symmetric kind of a benzene ring with nitro and amine groups. And then you've got melamine, less so damageable, but damageable nevertheless, which is used in Formica. So we actually damage them easily with X-rays. You can see the upper left picture. I have a crystal of, it's not a single crystal, but it's just a little bit of TATB explosive. It's not as big as it looks. That glass below it is just it's like a capillary, the thickness of a capillary. So we're looking at maybe 100, 150 microns. So don't worry, nothing blew up. But you can align the X-ray so precisely that black line is the X-rays going right through them to damage the TATB. So basically, based on my experience with damaging explosives as a function of energy with X-rays, I thought about the idea of doing the same with living compounds, living entities, for example, viruses. And so we actually have a patent for basically irradiating at a fixed energy, tuned energies, components, basically a virus, with the idea of, by knowing the spectrum, the X-ray damage spectrum, so to speak, let's say of DNA or RNA, you minimally damage the capsid, the outer part, which is what you want to minimally damage, but you selectively damage the interior based on this resonance idea. And so it's akin to if you're like an assassin versus, with a high-powered rifle versus somebody who's just shooting buckshot in a shotgun. You're sending all these X-rays out to damage everything in its path. What we're doing is we want to be the assassin and select certain components, certain metalloenzymes in whatever cells or viruses that we're trying to destroy. And why is that so much more powerful? Because we reduce collateral damage, and we don't need a very strong X-ray source to do it. So that was kind of my plug um, for the second part of this talk. So what are our future plans? Fluorescence detection is again related to the UV vis, and so that's a powerful method. And as far as detection, benchtop NMR suitability, I really would like to push the case for this. They're not that expensive from a DHS perspective. I mean, let's see, 5 years ago, I think one of those benchtop NMRs was about 80K. But again, for detecting certain types of species, I think it would be very powerful as a, once you get that spectrum, you know what it is right away. I would like to do one more visit of a DHS border facility. We're thinking of near Ciudad Juarez in El Paso, or the one in Nogales, Arizona. And then we're completing our design and then construction and demonstration of our portable remote UV-Vis and Raman system. So I just want to give a shout out to the people that worked with me, Jung Ko, JJ, Jung J. Ko. He's a technician in the chemistry department. Professor Corey Rusinek helped us with the cyclic voltammetry measurements. Drake Joseph, who still continues with me and was scheduled to go with me on this trip to Texas A&M, he's an engineer and physics major at UNLV. As I said, Nicholas Pudar has finished his degree at UNLV, and now he's in medical school. Kevin Ayala Pineda was an undergraduate who finished his senior thesis with me, and he is now a graduate student with me. And so he's being partially supported through this grant. And Angie Diaz Tremillo, she's a biophysics student. She's finishing her senior thesis with me right now. And this is related to instead of using X-rays, using electrochemistry to damage TMV, which she has successfully done. And we have some really exciting preliminary measurements. So in other words, instead of X-rays, we're using electrochemistry to damage these materials. And the argument is very simple. If you look at DNA, RNA in solution, it's an acid, so it's very conductive from a physics chemistry perspective. Whereas the exterior of proteins, for example, your skin, are not very conductive. So the argument is to use electrochemistry to, again, selectively damage the DNA, RNA inside whatever bacterium or mold or virus, so that you don't need to trash the whole thing. You're just basically trashing the interior, so it renders it sterile or inactive. And then Pravica Siflagu is a graduate student finishing his master's with me, and he's hoping to work on his PhD. And then again, I'd like to just thank everybody at Texas A&M and ORISE, Oak Ridge, starting with Jenny and Matt and Gregory and Chris and Cipra and Cedrica and Beth White at ORISE for all their help in what's been quite an interesting journey for us and continues thus far. So I guess I'll open up the floor to any questions. I hope we're still in. Can you all hear me?
Yes, we can hear you. I guess I can ask a question. This is Angela Irvin. I'm a DHS S&T. The Raman, typically you have issues with fluorescence, background fluorescence. How do you deal with that out in the field?
Okay, so first of all, we did have some issues. The most fluorescence was dealing with living things, like when we did the E. coli and the virus was extremely hard to detect. So actually, that told us that this didn't seem to be a chemical. The chemical fluorescence, we never really saw much of it. You may recall I showed some spectra of acetaminophen and whatnot. So we did not actually have a serious, you use background subtraction techniques. So these portable devices have their, you can automatically build that in. So in the field, we never, if it's a chemical poison, we did not really have much of a problem with the fluorescence. As long as you can see those peaks, you can identify the molecule. It's the living systems, the virus, the highly coupled systems that you have fluorescence. But then that tells me, oh, let's now maybe move on to UV vis, because this is not likely chemical. This is something that's likely more biological. Does that kind of make sense?
Yeah, you're using it to your advantage, actually. Yeah. Okay.
Yes.
Thank you.
Yes.
Yeah.
Yeah.
Michael, this is Greg. We had a couple questions that came in by chat, so I was going to ask one. The first one was related to the first part of your presentation. Would the portable Raman UV Vis tool provide identification, I guess basically have a library on board?
Yes. Yes. The UV Vis and the Raman and there were multiple systems. There wasn't just the one I showed you one. There's a nice one from Brooker, but it was a little more hefty, and I'm assuming the price would be hefty too. But they all had portable onboard, which could connect into a computer, but did not have to. They had their own screens. So right there, you get instantaneous or near instantaneous identification within your library of your sample.
Okay, great. And then I get there was a comment. You kind of mentioned borders as a singular term, but it's really any border anywhere, whether it's land, sea, or air would be fine for these kinds of tools.
Correct. I am, again, I can't solve the world's problems, in one day, but we were focusing on the land border only because, I know that the CBTS is very involved in that. But of course, all of these techniques would be just as valid whether you're on a ship or you're, a train or air. It's just the point is, the idea is to minimize the initial interaction with the officers as much as possible. I understand you can't do everything perfectly and Murphy will always take over, but the point is to have a remote system that maybe within a half a meter or a quarter of a meter, you can at least have this fiber, scan this strange powder that you see, so they don't have to touch it, because sometimes some of these things can actually go through gloves. And I know that from personal experience, not in a bad way. I just know it happens and I've seen it happen. So the idea is to have a remote detection that is in the field that gives them a very quick answer within a minute about that. Is this something chemical or is it biological?
Good. You're getting a lot of questions coming in. I'm going to try to get through a number of these. Another question was, how do you imagine using electrochemical techniques at the border? The implementation would be much more complicated than using ionizing X-rays.
Correct. And that's why I didn't really talk too much about it. Angie's not being paid off this grant, but it's actually very trivial, okay, because you have a bath. What we did is we had these carbon electrodes in a beaker, okay, and you just have a bath of, we had a buffer, and I'm sorry, it's a famous buffer solution, and I forget the name of it now, which shows you my, my ignorance of biology, but it was a phosphate, that's it, phosphate buffer, and we would flow a very reasonable amount of current. I'm talking less than an amp, and in the solution within 10, 15 minutes, and it destroyed many molecules. We looked at adenine, thymine, they all fell apart. We tried yeast, and we did this kind of a blue dye scan to sort of, and we call it amping when we flow the current through it, and then you can see how much you killed. So I'm not saying that electrochemistry is necessarily what the DHS wants. I'm just mentioning it as a possibility for if you have, I'm thinking you might have a bath of solution of this phosphate buffer. You dump something in there and you just irradiate it. Now somebody might say, well, you could add bleach. Yes, you could add bleach, but bleach is kind of toxic, and then you'd have to keep re-adding bleach and nobody wants to breathe bleach. Electrochemistry is just a very, less invasive. I like the X-rays the best personally because they just pass through and you're done. They don't transmute anything. You produce them. You just you basically inactivated whatever you have. Electrochemistry is more dirty, so you would kill it, but then you'd have to still deal with the consequences of getting it out of the bath, maybe cleaning the bath. So I just had mentioned that as kind of just a side. I didn't. That was not my original intention. I apologize about the electrochemistry. I just mentioned it something to think about.
Okay, great. Another question, kind of focusing on operations, kind of the designs, the technology used to detect biologicals. How would, in your description, a lot of times you talk about a powder on the outside of a box, but how about detection when you've got something inside a sealed container or luggage?
Yeah. So, again, when we were there at the San Isidro facility and we watched these beautiful X-ray scans, and what they do, and this is partly looking for dirty bombs. So that's very different physics from what we're talking about right now. That's nuclear physics, and they're looking for certain signature gamma rays or whatever. But they had an X-ray scanner that, some car was picked out, and they showed me scans. And for example, there was some poor guy that was hiding in some compartment. You could see it right there. So as far as a sealed container, the idea would be to look for something suspicious, look for something, what I call the delta concept, something that just doesn't look right. If there's a whole bunch of anthrax, if you've got a bunch of oranges in your crates, but then you see some sort of bag of powder, with an X-ray scan, then you might want to say, you know what, I want to look for that powder. Okay. Again, it's so, I'm not saying it's going to be, 100% effective technique, but in conjunction with something like X-rays, which can penetrate inside a something, and then you see something that looks out of the ordinary. Let's face it. All of these compounds have slightly different X-ray reflection, absorption, so you can tell that. We call it contrast. So, if you see something, again, I was focused on an example where we went into a trailer at Otay Mesa, and it was stacked with, I forget what it was, peppers or some crates of something. And I was just thinking, if an officer goes in there, first of all, it's kind of confining. If you're in a confined space, if there's something that is gaseous or, has spores, honestly, I was kind of scared. For the officers, and so the idea is at least if you see something, there's some crate, there's some package that looks out of the ordinary and maybe it has some trace powder on it, then you might want to investigate it further. Does that kind of help?
Sure. No, I think it's these folks that are kind of asking these questions are live and breathe a lot of this, and so I think the work's appreciated, but understood to be very complex. Especially as you're doing in a real world environment.
Yeah, I mean, one of the things that I learned from my visit to this facility was that, and again, I apologize. It's not my field. Every single, like they told me story after story after story. And then you realize that a lot of these solutions are not general. They are solutions for a specific threat. And so what you found is these experts, like Officer Yale, have decades of experience with all of these threats in kind of a global vision of what are we getting at the border. And so basically what they do is you kind of have this instinctive experiential memory within your institution, I would say even within each facility, where these experts are like, oh, I know what that is. That's such and such because we had this come in 1973. So, so again, I'm not solving the world's problems. I'm just simply saying there might be certain threats that this approach would work. Obviously, there are other threats. I mean, again, I just didn't understand. I remember there was an officer there, and he was picking apart some sort of a flower herb. I think it was an herb. And, and there was a small microscope. I mean, it just reminded me of something like I would give my students a project for this. Like, okay, we want to look for some sort of an insect or some sort of a contamination on this particular herb. And I mean, again, it's a specific herb. And like the officer Yale said, well, Mexico now, they're growing a lot of food for America. And so we're I mean, it's cross-border trade, which is totally cool, but we have to deal with these threats, which have accelerated because we have more of this trade going back and forth. And that's in fact why they opened up that Otay Mesa facility. That's a relatively new facility. So, I mean, again, you have a whole series of threats. We can't, there is no one specific protocol that's going to work for all the threats. So then you get in, and that's where the human being, the intelligence comes in, is that awareness of what are the possible threats that come in, and then what type of different approaches can we use? So all I'm suggesting, based on my observations, are that for certain threats where, I mean, again, it did happen. People sent powder in the mail and people died or got sick because of it, because it was anthrax. So I'm just saying, under certain circumstances, watching the officers work, and I'm not critiquing them. I understand they have a very hard job, and they've got thousands of, I saw the trucks, there are hundreds of trucks backlogged and everything. But the point is to at least initially reduce that possibility of some sort of an exposure using a remote technique, I think is a good approach. And I'm just saying, I didn't see that at the Otay Mesa. I didn't see that at the San Ysidro. I did see a lab. I did see a portable Raman spectrometer in the lab. And we got talking shop. I got into, where do you get your solvents from? Where it was a total lab. But the point is, I think it would be helpful for officers to have something when you open up that truck right away and you don't know what's in there, and you don't know what you're breathing. I just think it would be helpful to have some sort of a remote detection device right then and there that might save them from exposure.
Great, thank you. We only have a few more minutes. A couple things. One, we went over it at the beginning, but just to remind folks that we've made a recording of this presentation. Michael, will your slides be available? Could you send them, for example, to us, or just would you prefer folks to reach out to you directly?
They can reach out to me, or I'll be happy to send you them as soon as I finish the talk today. Okay,
and then the other question is, there was some interest in sharing these with folks outside of DHS. Is there any problem with that?
No, not at all. I'm trying to help our country, so it's fine. It's not. I'm not in it for the money. I'm in it to make a difference.
Great, thank you. And then a question came up about the kind of the damage that's done. That we've all seen. Whatever you, especially when it was my USDA days, the letters and cards that would come through the X-ray that would be so damaged or brittle that they were useless. when you were describing the X-ray techniques, that was really to do damage, right?
Correct.
Not necessarily to just for surveillance of any sort.
No. That was actually to do. Now, that's the X-ray. Yeah, the X-ray techniques. Now, let me just clarify that because of the essence of the tuned X-ray technique, the monochromatic assassin's bullet technique, you're not going to yellow the whole thing. You're just focusing on certain metalloenzymes or certain structures in these cells or viruses that are particularly that have an enhanced absorption. And so I'm not saying there's no damage. I'm just saying there's going to be a lot less damage than the conventional techniques.
Okay. And that goes to kind of one of the first questions that I skipped over, but I'll get back to. In the case of irradiating viruses, given that they replicate inside liquid media, so to speak, how would you achieve the level of damage for a particular viral type, considering that the radiation has to pass through cells first?
Well, again, if you're dealing with, as I mentioned, the 25 keV, we call them hard X-rays. So these are much more penetrating X-rays, and basically they will penetrate up to a certain point. I mean, it depends on, of course, how big your sample is. I was in the viruses. I wasn't necessarily thinking of a fluid. Of course, I understand they might be connected to a fluid, but like an airborne situation where you're just irradiating, they go through water very easily. The only issue is if you have heavy metals, then those hard X-rays get absorbed, which is exactly the point I'm trying to make. All living systems have heavy metals, or less heavy metals. So let me give you an example. Zinc. What does everybody say when you're sick? Take zinc, okay? The K edge of zinc is just below 10 keV. So the point is, if you can use my technique, there's zinc all over your cells, and even in some viruses, in these metalloenzymes. And so the point is, if you can irradiate at 10 keV, whatever's around that zinc gets destroyed, but the rest of the stuff is fine. So 10 keV will get through. It doesn't have the greatest attenuation, but there are heavier metals, okay, that can be used that will have better attenuation. Lead, of course, is one of the best. I mean, that's over 80 keV. So, but the point is, the X-rays will get through. Hard X-rays get through water up to a certain point, and then you use this resonant absorption to destroy whatever's near those cations. You're good to go.
Excellent. Well, we are almost at the limit of our hour, and I want to thank you on behalf of CBTS, but also all of us that are involved in any kind of Homeland Security activities for the work that you're doing and your willingness to kind of cast the net to search for new solutions, at the same time kind of grounding it in some of the realities that you've observed. So thank you very much. Again, we appreciate if we can have folks get a hold of you as they need. We'll make sure that your email address is available on our website with the slideshow. So if there's anything that you want to add that you might have forgotten, certainly feel free to add that at either at this point or in anything you might want to send us afterwards.
Well, let's put it this way, Greg. I consider this an initial progress report talk. I'm hoping when I heal, hopefully by this spring, I'll make a real visit to your facility, and then we'll give the sort of the final, the finish the second time. Okay.
Wonderful. Well, thank you, and I want to thank everyone that joined us today. We look forward to having you come back. We've got a number of seminars that are on the docket. The next one is November 15th, and then we have one following up on December 1st, and we'll make sure that we get those announcements out. But Michael, thank you very much, and all the best in recovery here.
Thank you very much, Greg.
All right, take care.
Take care, everyone. Okay.
Thanks.
Bye.
Bye-bye.
Bye.